I think Andy did a very good job at introducing my next talk, about how actually microplastics could affect humans, and should we be concerned about the implications of microplastics and human exposure. So I'm going to talk through all the different topics related to microplastics and humans, and hopefully we can come up with some questions at the end that we can all discuss together. So as we just heard, microplastics have been found throughout the environment and ingested by different animals, and the toxicological and biological implications of microplastic ingestion or exposure are not completely understood. We have started investigating, and there's ongoing research, but we really haven't got that much information in regards to biota. And well, we don't really know much about for humans either. So I am going to go into as much detail as I can from the research that's available and discuss ways forward to investigate the potential implications of microplastics on humans. And I must mention that we know a lot about microplastics, but we still don't know enough about the nanoplastics. And it's quite possible, as you'll hear from the rest of the talk, that it's the nanoplastics that we should be more concerned about when it comes to human exposure. So there's multiple different routes that humans can be exposed to plastics, and I will go through all of them. But as an overview, we can be exposed to plastics orally through intaking plastics in our foods, through inhalation if we breathe in microplastics that could be present in the air, and also through skin exposure, so through dermal exposure if microplastics are on our skin and they cross the dermal boundaries. And to discuss these three areas, we're going to go through firstly consumables, dermal exposure, inhalation of plastics, and then what this means on the impact of human health and the potential indirect effects that could come from plastic exposure. So before I start on consumables, you probably have seen recently in the media quite a lot about microplastics being in drinking water. And this was a study that was conducted for Orb Media that hit the headlines around the world. And indeed, many of us researchers in Norway were getting phone calls to find out what was happening in Nordic waters and what our opinions were on this study. And I think it's significant to note that plastics were found in tap water, although the values that were reported were variable, and we should probably look at it in much more detail. So I'm going to present the results here for you, just so you can get an idea that where plastics are in tap water. So the study was conducted globally. There were 159 samples collected from 8 geographical locations, and from 14 countries, it was 83% contamination, and it ranged from low numbers of particles, but the average was 4.34 particles per litre. So plastics were present in drinking water. But what does that mean? And there weren't really any significant differences around the world, as you can see from the figures here at the bottom. The percentage ranged from 79%, 74% up to 94% in the USA. As to why this plastic contamination varied between countries, there could be a number of reasons. It could be the method of collection to start with. It could also be the water processing procedures, and anywhere along the pipelines from where the water is produced to the tap. There's multiple sources of contamination. And this, I think it's important to note that this is, it's not that we, plastics have probably always been in our drinking water and drinking sources, and it's just been highlighted recently because of the focus on microplastics. So there's been now lots and lots of, I suppose, it's lots more attention from media, from the public, because we've become more aware of microplastics' presence. But they've probably always been here since we started producing them and they started breaking down in the environment or through production. So I don't know we just need to understand that plastics are there to start with, and our next steps is to find out what this could mean. But before we can do that, we thought we were approached actually at NIVA to find out what was happening in Norwegian waters. And I don't know if anyone saw the newspaper before Christmas in Vega. It was all over the front page news that microplastics were in drinking water. And I'm going to present the raw results here for you today so that you can have a look at where Norway stands in comparison to the global average that was presented by Orb Media. So we looked at 11 different sites around Norway, and the sampling method was exactly the same as the Orb Media report because we wanted to compare our results directly to the global study. We collected the water directly at taps, so drinking water locations in people's houses around Norway. We got 3 replicates from each location so that we could see if there was differences between our samples, and we conducted our analysis in our controlled laboratory. So our methods were as controlled as they could be, although we do highlight that there could have been some limitations within the sampling process because the water samples were collected by different individuals, but they had very strict protocols to follow. And they were all NIVA scientists or representatives around Norway. Actually, we had the town mayor, the mayor of Oslo collect the ones from the Rådhuset. So that was interesting. So if we look at the results for Norway are the black and white squares. So the black squares are the microplastics less than 1 millimeter in size, and the white squares on the top of the bars are the plastics that were greater than 1 millimeter. So we could include both the size definitions that we have already discussed. And you can see actually the levels of contamination were quite low. And the numbers really ranged between 0 and 2. There were not many particles in any of our samples, which is much less than the global average. So we didn't find as much as around the rest of the world. And I was actually quite shocked that I found any particles, and our limits of detection were we're in a range where we said actually the conclusions we can draw from this is that we need more samples. We need larger volumes of water because the volume of water we were using to collect our samples was 1 litre per replicate, and that's not enough for drinking water. If we used 1 litre of water from influent to a wastewater treatment plant, for example, we would have needed much less water because it's heavily contaminated. But drinking water has been through several stages of purification, which is probably why we got very low values. And we want to repeat the study, and in fact we are repeating the study with much greater volumes of water and more sites around Norway on behalf of Norsk Vann. So we are going to investigate this further. So this is the preliminary data. And we want to go further and understand what is happening with plastics in drinking water. The next step that we could be exposed to plastics orally is through seafood and ingesting contaminated food, as Andy has already touched on. And this is a question I get all the time as to how can we be affected by eating contaminated seafood. So I'll just show you a little diagram. This isn't Brian, this is probably Brian's friend. I haven't got a name for him, I'm afraid. He is seafood. He's a langoustine or Norwegian lobster. And then we have some fish too. So both these are seafood that we eat and we consume regularly. And the question is, do they result in plastics on our plate? But for these species, and they are examples, they are processed where we remove their digestive tract. So the only part of the animal that we are consuming is the flesh and the tissues. So it's quite unlikely that there's going to be microplastics in the tissues because microplastics can't pass out of the gut walls because they're too big. They can't cross the cell boundary. So in respect to plastics being on our plate when we're eating a portion of mackerel, it's probably very unlikely, unless plastics have arrived there during the filleting process on the kitchen table and airborne fibres have landed on a fillet of fish. But if we talk about the mussels, it's a different story. As Andy said, we eat mussels and other shellfish whole, other bivalves whole. So if there is plastics inside the digestive tracts of these organisms, then yes, it is possible that those plastics will be transferred to us. But once they've been transferred to us, what does that mean? If they're microplastics, it's probably very likely they're just going to exit our body. Now, I use this, probably you might laugh, I chew my pen all the time. I've been chewing my pen since I was a child, so I've probably been ingesting lots of microplastics my entire life, and I haven't noticed any effects yet. So, and I'm pretty sure that when I used to swallow plastic toys as a kid, they all came out the other end. So that's how I look at microplastics at the moment, to try to understand that they will eventually get through the body unless they get stuck. And we've heard that chewing gum can remain in your stomach for 7 years, which is no longer true. But that used to be the story that went around at school, so you never wanted to swallow your chewing gum because it would stay there. But it's much quicker for it to pass through your body. Our bodies are quite sophisticated in that respect. But if we do return to the bivalve consumption, as I said, plastics can be in bivalves and they could enter our food chain. But I want to show you some maths that we conducted for a report for the Food and Agricultural Organisation. We took a portion of mussels that was 225 grams, and we assumed that there were about 30 particles of plastic per mussel based on the relevant literature that was available at the time. This would work out to be about 900 particles in the one meal. And if we assumed an average size of the plastics to be around 25 microns, and we took an average density, it was polyethylene we used, so it was 0.90 grams per centimetre cubed, that would result in there being 7 micrograms of plastic in the portion of mussels. If we compare that to an average human being, it's 0.1 micrograms per kilogram body weight of a 70-kilogram human being, which is very, very low. But what does that mean? We've discussed that plastics can harbour chemicals, potentially through absorption and additives that are incorporated. And it turns out that the plastics would contain 0.1% of the total dietary exposure of any chemical compounds, which would lead us to say that microplastics contribute to a very small fraction of the total dietary intake of contaminants. And there are many other sources of contaminants available to us. I won't go into them, but we should also consider the other routes of health implications rather than just microplastics. Other forms of consumables that have been found to contain plastics include honeys and sugars. Fibres and fragments have been identified, and you can see from these values 174 fibres per kilogram of honey and 217 fibres per kilogram of sugar. And sea salt as well, mainly polyethylene and polyethylene tetraphthalates contained microplastics. These were greater than 45 microns in size. And for those of you that like alcohol, out of 24 different samples of beer, there was less than 109 fragments per litre. These were all preliminary studies, and more in-depth investigations are required. Now that the technology and the methods of detection for microplastics have advanced, the studies are probably going to be repeated because there's more ways to control for contamination. We've become more aware of the way that researchers could contaminate their own samples. We've also got much better identification techniques to allow us to look at plastics a different way, looking at different sizes and different ways of classifying them. So I would urge everyone to continue eating honey, sugar, salt and beer, because we need more information before we say we can't drink it. So, summary, consumables, yes, we can be exposed to microplastics through eating food. At the moment, I don't think it's necessary to stop eating and stop drinking. We are probably going to be exposed to more severe chemicals or other forms of contamination in the long run. We should just be aware that we do know that plastic contamination is going to be increasing in the environment, And if plastic contamination is increasing, it's more likely that animals are going to become contaminated with plastic as well. So we should just bear in mind that these microplastics are out there. The next form of interaction on potential human uptake is through dermal exposure. So this figure here is are microbeads that are incorporated into body scrubs and other cosmetics. They can vary in size, but most of the body scrubs contain plastics that are greater than 100 nanometers. And because of that, there's going to be no effect on dermal exposure because they can't be absorbed through the cell boundary. However, smaller fragments, the nanoplastics, have the potential to penetrate the skin, and therefore they can cross the cell boundaries, and we might see uptake there. But as of yet, I don't know about nanoplastics in cosmetics. As far as I'm aware, they're removing most microplastics, so we shouldn't see nanoplastics. It could be the case that if nanoplastics are airborne, that they touch your skin, they could be absorbed, but we have no information and no data to back this up. This is just speculation as to what could happen, because we are only now starting to look at the human effects of plastic and microplastic exposure. So I've been talking about atmospheric airborne particles. This value on the screen was taken from Paris in an urban area, looking at atmospheric fallout of airborne plastics and airborne fibres. And the values range between 29 to 280 particles per meters squared per day. And that's so that we know that plastics are airborne and they're around us. They're going to be floating in the air that we breathe. On a good day when the sun's shining through the window, you can see particles in the air, dust particles. And plastic fragments and microplastics are a part of the dust particles that you see. And that means that if we're breathing the particles, we could be taking them in. These could be particles that are produced from wear of clothing, wear of tires, wear of city dust, produced from industry, but also the breakdown in the environment. And it's significant that we need to understand what these plastics could have, the effect these plastics could have on humans, and what could happen once the plastics are breathed in. So that will bring us on to the impact on human health. So we said plastic can be breathed in through inhalation, and how does it affect us? We have seen some studies that identified synthetic fibres in the human lungs. So it is correct to say that plastics can be breathed in, or fibres can be breathed in. But if you look at the sizes on the figure there, the size of the plastic will be stopped at certain areas. So plastics greater than 10 microns will probably not get any further than the nasal cavity because of the membrane and the mucus filters in your nose. But also when it enters the trachea, it can't pass the boundaries of the trachea at 5 microns, linking down to the bronchus. And all the way when we get to the alveoli, and this is where we're looking at where blood and air are very close in contact. I have not done biology for a long time, or human biology, but this boundary is so close that the anything less than 1 micron could reach the cell boundary where they can interact. So we're looking at nanoplastics by this point. So the microplastics have a limited route through the body. It's going to be the nanos. And so as Andy highlighted earlier with Brian, that it's the size and the exposure route that is more important, and it's more significant that the smaller we get, it has the greater potential to have an effect. The human body has several different ways it can deal with materials that it doesn't want to have that it doesn't want inside of it. We have mechanical clearance mechanisms such as coughing and the mucus escalator, which removes particles from our trachea when they're an irritant. When we're coughing, it's because something's causing an irritation, and it allows us to expel them. Phagocytosis is when cells take in the particle to remove it from the situation. And also we have lymphatic transport, again, to remove particles. So the body is very capable of dealing with items and materials that it doesn't want. So it has the opportunity to remove it. But we also need to consider how plastics can be deposited. If they escape the clearance mechanisms, they can cross the cell boundaries in the form of diffusion. They can lodge themselves into different parts of the I've forgotten the name of the word, the cells on the cell linings, and they can also cause blockages and go across that way. So, and if there's a large amounts of them, they could cause impaction, which is the blockaging of the airways. But it's most probable that you have particle deposition and clearance. So we need to understand these mechanisms a lot more. In regards to microplastics and nanoplastics, we really don't know. We haven't investigated it enough. And if we move on to the next page, what are the consequences of inhalation? There's a really good review that I've been using the figures from, and it looks at the potential implications of microplastics inhalation. And if plastics do cause irritations and they cause immune responses, it could lead to airway diseases, interstitial lung disease, and potentially cancer. But we still don't have enough information to take this further. These are all still areas that are under investigation. But because plastics do have low toxicity, rather than causing chemical responses, they could be the responses that are from when we get rubbing and causing inflammation. If an individual maybe has a lower immune system, a reduced immune system, then these responses might be higher. But again, we still don't know. There's lots of ifs and buts and maybes going on, but we really do need just to consider the ideas of what's out there. And the microplastics researchers, I will mention, most of us are all marine biologists, and that's where the research started. So we've started to branch into different research areas and bring in experts from different research fields, which has led us to expand our knowledge base and also start moving into effects such as what are the potential human effects. So that's inhalation, but what about in the consequences of ingestion? And these are very complicated images, so you don't have to look at them if you don't want to. But I just wanted to say that if we're taking plastics in through food, through drinking water, that if plastics enter our digestive tract, what happens? Well, there's very, very few reports of the translocation of plastics from within the gut to outside of the gut. Although, if particles are less than 130 microns, they can transfer into the lymphatic system. So there has been some mammalian studies that show that. And less than 110 can be the transfer from the portal vein into the liver. And now these are mammalian studies based on dogs and rats. So the potential effect and the potential toxicity of this is that there could be the physical presence of plastic within the gut, causing blockage. But the only in vitro study actually on human health, it was looking at reactive ROSs, so reactive oxygen species. Oxygen species. And it's looking at the generation of them in the cerebral, but also in epithelial human cells. But that was focusing specifically on polystyrene nanoplastics that were at 40 and 250 nanometers. And the exposure level was higher than what we're expecting to see in the environment. So it was more of it was a toxic study to see if there was a response. Again, we need to go back to it's the size and the exposure route that will influence potential risk to human health. And that's why I keep mentioning nanoplastics, because microplastics are probably too big to cause any effect, but the nanoplastics have a greater chance of causing something. We also need to consider the physical presence of plastics. So they lead to blockages and inflammation. And so here you can see the endocytosis gut uptake. The particles need to be less than 10 microns. So the immune response, which I just mentioned. And this is paracellular presorption, which would happen in the gut lining, and the movement of particles into the lymph vessels across the gut. But we're still investigating it, and research is ongoing. So that's about it that I can give on that topic. So what about the effects then? We know that we are exposed through different ways. But what are the indirect effects? And as Andy has already mentioned, we were talking about that plastics can leach additives that are incorporated into plastics during their production. So these include the PBDEs, phthalates, PAHs, bisphenol A, etc. Once plastics, we've seen that chemicals will migrate from macroplastics into food. For example, sometimes plastics can leach chemicals into food products. That's why BPA was banned from babies' bottles. But this is the macroplastics. So there used to be, there still is, a huge concern about the chemicals that are used to make macroplastics and how the chemicals can leach out. I think one example is if you heat a plastic bottle, if it's got sometimes metals in it, between 60 to 85 degrees, you can see the leaching of these chemicals from the plastic into water or whatever liquid is inside. And that's why you're not encouraged to use plastics in microwaves or only certain microplastics, because chemicals can be released. And they can be a major route for human exposure. But these are the macroplastics, not the microplastics. So the role of microplastics and the leaching of additives has been raised, but it's quite possible that there are other sources which have equal or a greater importance for chemical transfer, such as leaching from larger plastic items. And I think that's important to note, that we can't forget about the large items of plastic. All of research at the moment seems to be going into microplastics, but macroplastics also have an effect too. And the last one is looking at the absorptions of contaminants and as vector pathogens on microplastics. So in a very most simple form I could manage, because microplastics have a very high surface area to volume ratio, and they're hydrophobic, which means that they can absorb contaminants from the environment. And if these plastics are in the environment, they've absorbed chemicals, they can transfer into food chains if animals eat them, and there's a high enough level of chemical on the plastics. There could be a movement into the lower trophic levels. But we could also see biomagnification to predators, and this is where the chemicals then reach much higher numbers in animals. But I'm referring to the chemicals themselves and not the microplastics, because if the chemicals leach, once they're on the plastic and then they're inside an animal, the chemicals can leach and then they can move into the next organism. And we have seen biomagnification of different chemicals in animals around the world, but we haven't seen biomagnification related to microplastics yet. It has been assumed, but we haven't actually seen it. We haven't been able to directly monitor it yet. Although research, again, is underway, and I'm sure in the next few years we'll have more answers. Currently, we have more questions, and we just have to keep going, and we will eventually get there. Just give us some time. So, in summary, we need to consider all routes of microplastics and their associated chemicals, and other sources of microplastics and their associated chemicals, and not just drinking water and food. And we need to think about how we can be exposed through breathing or or being exposed through the skin. It's not only seafood that could be exposing us, but we also have many, many unknowns, and we will try and answer as many questions as we can, but it's going to take us time because we're still identifying the most appropriate methods to allow us to identify and understand the implications of microplastics. Most of the time, so we need to understand exposure concentrations from dietary as well as airborne sources, but we still really don't know what happens in humans. We're getting there, but we just don't know. We also need to consider the cellular mechanisms that could go on with microplastics and nanoplastics, and we need to understand what the toxicological response is. We've started understanding the toxicological responses in marine organisms, but we're far from understanding them in humans. And we also need to consider size and shape of plastics. Most of the studies that are conducted at the moment use polystyrene beads, and we need to consider the other forms of plastics that are in the environment. So on that note, I'll say thank you for listening to me twice, and I'm sure that myself and the other speakers are going to be discussing with you later on some questions.